Good morning, everybody. Welcome back to Daily Lectures by New Age Cosmetic White, EdTech Healthcare Consultancy. I am Dr. Bobby. We are on the subject of molecular biotechnology, and specifically on to Chapter 20, which deals with engineering plant quality and proteins. In continuation with yesterday's lecture, and I'm on to the topic about the specific topic of the flower industry that is continually attempting to improve flower appearances in post-harvest lifetime. By traditional breeding techniques over the years, it has been possible to create thousands of new art varieties that differ from one another in color, shape, and plant architecture. However, traditional plant breeding is a slow and painstaking procedure that is limited by the gene pool of a particular species. Thus, for example, no one has been able to breed a blue rose. As an alternative to traditional breeding techniques, uniquely colored flowers can be developed by manipulating the genes for enzymes in the anthocyanin biosynthesis pathway. Anthocyanins, which are a class of flavonoids, are the most common of flower pigment and are the major constituent in orange, red, violet, and blue flowers. They are synthesized from the amino acid phenylalanine by a series of enzyme catalyzed reactions. The color of the flower is determined by the chemical side chain substitutions of different chemical structures, for the cyanide derivatives producing more red and the delphinidin derivatives producing more blue. The color of the flower is determined by the chemical side chain substitutions of different chemical structures, with the cyanidin derivatives producing more red and the delphinidin derivatives producing more blue. Moreover, plants sometimes contain both flavonoids and carotenoids. It is the combination of the two that produces the wide range of colors seen in nature. While the Petunia enzyme dihydroflavanol 4-reductase can convert colorless dihydroquercetin to red cyanidine 3-glucoside and colorless dihydromycetin to blue delphinidin 3-glucoside. It cannot use colorless dihydroteamferol as a substrate. However, when petunias were transformed with the dihydroflavanol 4-reductase gene from corn, the flowers of the transgenic plants were brick red orange. This unique color, which had never been seen before in petunias, was due to the production of pelargonidine 3-glucoside by the transgenic plants. Moreover, following laboratory manipulation of transgenic flower pigmentation of subsequent field testing in 1996, the company Florigene introduced moon dust, a mauve carnation, into the marketplace, followed by moonshadow, a violet carnation. In 1998, conventional breeding had failed to produce these flowers with hues in the mauve, blue, violet range because they lacked the ability to produce the blue pigment, delphinidin. 4 additional varieties of carnations that feature different tones of violet and blue have been added. To date, over 75 million of these flowers have been sold worldwide. By mid-2009, several uniquely colored transgenic roses were undergoing field trials and were expected to be available to consumers around 2010. More than 24 field tests with the new designer plants have been permitted. Among them are light blue Terrenias, bronze-colored forsythia, and yellow petunias. One recent studied the production of anthocyanin pigment 1 PAP1, mauve transcription factor from the plant A. thaliana, was stably introduced into petunia plants, Petunia hybrida. This transcription factor is known to regulate the production of non-volatile phenylpropanoids, including anthocyanins. Surprisingly, in addition to an increase in pigmentation, backbone transgenic petunia flowers demonstrate very large increase in the production of volatile phenyl propanide benzoate compounds. This coordinated regulation of petunia flower color and the scent production by backbone provides a clear advantage for plant survival in terms of attracting pollinator insects. In addition, this work suggests a strategy to create flowers with both novel colors and enhanced scents. The carotenoid astaxanthin, which provides a characteristic pink color to salmon, trout, and shrimp, is synthesized by marine bacteria and microalgae and then passed on to fish. Should the food chain more important, astaxanthin protects salmon and trout eggs from damage by UV radiation and improves the survival and growth rate of juveniles. Most likely, these properties of astaxanthin are related to its function as a powerful antioxidant. However, when fish are grown in aquaculture, they are separated from the natural food chain, and astaxanthin must be added to their feed in order to impart the typical pink color to their flesh. Currently, astaxanthin is chemically synthesized and accounts for approximately 15% of the total cost of salmon farming. To produce astaxanthin biologically, one group of researchers first cloned the cDNA, encoding the enzyme beta-carotene catalase beta C4 oxygenase from the unicellular green alga Hematococcus pluvialis. When this cDNA was expressed in tobacco plants that contain beta-carotene and the gene for beta-carotene hydroxylase, astaxanthin was synthesized. By appropriate genetic manipulation, astaxanthin has been synthesized in tobacco flowers. The cDNA for the algal beta-carotene catalase was fused to DNA, encoding a chloroplast transit peptide, and is used to transform tobacco plants. To limit the expression of astaxanthin to flowers and fruits, the cDNA for the algal beta-carotene catalase was fused to the promoter of the tomato PDS gene, which is actively produced primarily in tomato reproductive tissues. To increase the expression of cDNA for the algal beta-carotene catalase, the DNA fragment carrying the PDS promoter was modified by deleting portions of its DNA sequence and then fused to a beta-glucuronidase gene. The construct, which had a 305BP deletion, I repeat, deletion from the 5' terminus of the PDS promoter, showed a decrease in beta-glucuronidase activity in leaves, sepals, and petals, and a very large increase in activity in flower ovaries and nectaries. To obtain maximal gene expression ovaries, a 305BP deleted PDS promoter was placed upstream of the cDNA for the algal beta-carotene catalase. The net result of all these genetic manipulations was that once the introduced strands of DNA, tDNA had been inserted into the plant genomic DNA, the algal beta-carotene catalase together with a transit peptide was inserted through the chromoplast membrane, with the transit peptide being removed in the process. Once inside the chromoplast, the algal beta-carotene catalase worked in concert with the endogenous beta-carotene hydroxylase to convert the beta-carotene to astaxanthin, which accumulated in the flower nectaries. The advantage to producing astaxanthin in plants rather than bacteria or other microorganisms, the plants can store large amounts of carotenoids inside cells and lipids vesicles within the plastids. These plants can accumulate 10 to 50-fold higher concentrations of carotenoids than microorganisms whose membranes are damaged by high concentrations of carotenoids. These manipulations have the potential to dramatically lower the cost of the astaxanthin that is used in salmon farming. Plants as bioreactors. Plants are easy to grow and can generate considerable biomass. With these features in mind, research has been carried out to determine whether transgenic plants can be used for the production of commercial proteins, chemicals, and like recombinant bacteria They're grown in large bioreactors, a process that requires highly trained personnel and expensive equipment. Crops can be produced relatively inexpensively by less skilled workers. In addition, when proteins that are intended for human use are produced in transgenic plants, there is a significantly reduced risk of mammalian virus contamination in comparison to proteins that are produced in animal cells grown in culture. Ultimately, the biggest hurdle to overcome in the production of foreign proteins in plants is the purification of the product of a transgene from the mass of plant tissue. On a laboratory scale, plants have been used to produce monoclonal antibodies and antibody fragments. The polymer polyhydroxybutyrate, which can be used to make biodegradable plastic-like material, and a number of potential therapeutic agents and vaccine antigens. Antibodies. The production of antibodies and antifragments in transgenic plants has several potential advantages over the synthesis in recombinant microbial cells. For example, transformation of plants generally results in the stable integration of the foreign DNA into the plant genome, while most microorganisms are transformed with plasmids that can be lost during a prolonged large-scale fermentation. In addition, the processing and assembly of foreign proteins in plants are similar to those in animal cells, whereas bacteria do not readily process, assemble, or post-translationally modify eukaryotic proteins. Moreover, plants are inexpensive to grow on a larger scale, and their production is not limited by fermentation capability. It is estimated that it costs approximately $5,000 per gram to produce antibodies from hybridoma cells in culture, $1,000 per gram to produce antibodies from transgenic bacteria, and $10 to $100 per gram to produce antibodies for transgenic plants. However, since most harvested plant tissues cannot usually be stored for long periods, foreign proteins might be produced in seeds, where they will be stable for long periods under ambient conditions. To date, a large number of antibodies, including immunoglobulin G, IgG, IgM, single-chain Fv fragments, and Fab fragments, have been produced in plants. Some of these plants produced antibodies, sometimes called plantibodies, have been purified and used for diagnostic and therapeutic purposes, and others have been used to protect the plant against certain pathogenic agents such as viruses. In a small number of instances, plant-produced antibodies are being tested in clinical trials to determine whether they are essentially equivalent to antibodies produced in other host cells. Effective industrial-scale production of antibodies synthesized by plants has until recently been hampered by a very low yield, typically in the range of 1 to 10 micrograms per gram of fresh biomass. Moreover, the conventional transgenic plant technology, it is estimated that it takes about 2 years from the beginning of the cloning process to produce the gram quantities of antibody. But it is still problematic to coordinate the synthesis and assembly of the two different polypeptides that are integral components of antibody molecules. Moreover, traditional transgene expression systems are limited by the low infectivity of the viral vectors, especially those carrying medium-sized to large-sized inserts. It's also necessary to ensure that transgene expression systems based on viral vectors do not spread to plants other than the intended host. This is typically done by using mutant plant viruses that are unable to produce capsid coat proteins and therefore cannot form functional viral particles. However, these systems generally produce only very low levels of antibody. Fortunately, an alternative transgene expression system has been developed. This expression system involves co-infection plant cells with the two separate plant virus vectors, one based on tobacco mosaic virus and the other on potato virus X, so that the two vectors do not compete with one another, but rather can coexist within the same cell. These vectors co-replicate within the plant, with each vector expressing a different antibody chain. That is, one expresses the light chain, the other expresses the heavy chain. This system has been used to increase the amount of IgG antibody that is typically synthesized by a plant transient expression system by about 100-fold, so that it is possible to produce up to 0.5 milligrams of assembled IgG antibody per gram of fresh leaf biomass. At this high level of expression, it is possible to grow transgenic plants that produce specific IgG molecules in small areas indoors in controlled greenhouses, thereby avoiding environmental concerns regarding the inadvertent escape of antibody-producing plants. The potential of this technology provided the incentive for the sale of the company, where it was originally developed, to a larger company that hopes to open a clinical-grade manufacturing plant with the objective of beginning clinical trials of the antibodies produced. When known to polymers, it is costly to produce the polymer poly-3-hydroxybutyric acid, which is used in the synthesis of biodegradable plastics. Which is done by bacterial fermentation. Consequently, research has been conducted to determine if the polymer could be produced at a lower cost in plants. And bacteria, such as alkalogens, eutrophs, poly-3-hydroxybutyric acid is synthesized from acetyl coenzyme A in 3 steps catalyzed by 3 enzymes. The genes that encode these enzymes are organized on a single operon. Since plants are unable to process the transcript of an operon with more than one gene, each of the 3 genes was isolated and cloned separately into a plasmid. The genes were targeted to the chloroplast of the plant Aethalia because previous experiments had demonstrated that cytoplasmic synthesis produced only low levels of the polymer, and the transgenic plants were highly stunted. Moreover, chloroplasts can accumulate high levels of starch and other biological polymers, so it was thought that they would similarly be able to accumulate large amounts of poly-3-hydroxybutyric acid. Unlike highly valued proteins that need to be produced high levels in plants for production to be economically feasible. Each of the 3 poly-3-hydroxybutyric acid biosynthesis genes was fused to a DNA fragment that encodes the chloroplast transit peptide of the small subunit of P-ribulose biphosphate carboxylase, and was placed under the transcription control of the cauliflower mosaic virus 35S promoter. Separate plants were transformed with each construct with typlasmid binary vectors. 2 transgenic plants, each with a different foreign gene in its genomic DNA, were crossed to form a transgenic plant with the 2 foreign genes. Then the double genic transgenic plant was crossed with a transgenic plant that carried the 3rd foreign gene, and a transgenic plant carrying all 3 of the bacterial poly-3-hydroxybutyric acid biosynthesis. Genes were selected. Mature leaves of some of the triple gene transgenic plants produce more than 1 milligram of poly-3-hydroxybutyric acid per gram fresh weight of leaf. Unfortunately, Aethalia plants that produce very high levels of poly-3-hydroxybutyric acid were severely stunted. Nevertheless, this work is the first step in the development of plants that produce large amounts of poly-3-hydroxybutyric acid. However, to realize the commercial potential of the system, these polymers It will have to be produced on plants with other than the Aethaliana, so that there will be a much greater amount of plant biomass produced. Edible vaccines. Although considerable progress has been made in recent years in the development of new vaccines in many countries, either the vaccine itself is too expensive to be used on a large scale, or there is a lack of physical infrastructure. For example, like roads and refrigeration that makes it impossible to disseminate the vaccine. Commercial vaccines are expensive to produce and package and require trained personnel to administer injections. Clearly, it would be advantageous if vaccines could be delivered inexpensively on a broad scale in an edible form. Example, as part of a fruit or vegetable, when vaccines are taken orally, they can directly stimulate the immune system. An edible vaccine, in contrast to traditional vaccines, would not require elaborate protein production facilities, purification, sterilization, packaging, or specialized delivery systems. Moreover, unlike many currently utilized recombinant protein expression systems, plants' glycosylated proteins are a factor that may contribute to the immunogenicity and stability of a target protein. Much of the work on edible vaccines has been reported so far utilizes potatoes as a delivery vehicle. Potatoes were originally chosen for this work because they were easy to manipulate. And however, potatoes were never intended to be the vaccine delivery plant. They require cooking to make them palatable, and cooking destroys and activates most protein antigens. Plants that are being considered for the delivery of edible vaccines includes bananas. All the banana trees require several years to mature. Tomatoes, although tomatoes spoil readily. Lettuce, carrots, peanuts, and corn, mainly for vaccinating animals. Cholera is an infectious diarrheal disease caused by the enterotoxin produced by the gram-negative bacteria Vibrio cholerae. Since Chapter 12, globally, there are more than 5 million cases and 200,000 deaths from cholera each year. To test whether it might be possible to develop an edible vaccine against V. cholerae, potato plants were transformed using a tumefacients with the cholera toxin subunit B gene. Cholera toxin subunit B binds to an intestinal receptor. Subunit A contains toxin activity. 1 gram of transgenic potato produced approximately 30 micrograms of subunit B. After the transgenic potatoes were cooked in boiling water until they were soft enough to be edible by humans, approximately 50% of the subunit B protein remained undenatured. The cooking potatoes were fed to mice once a week for 4 weeks before the mice were tested for antibodies against the subunit B protein and for resistance to V. cholerae caused bacteria. These tests indicated that the mice had acquired a significant level of protection against V. cholerae. Now, the cooked potatoes were fed to mice once a week for 4 weeks before the mice were tested for antibodies against subunit B protein and for resistance to V. cholerae caused diarrhea. These tests indicated that the mice had acquired a significant level of protection against V. cholerae. Moreover, all the mucosal antibodies titers declined gradually after the last immunization. They were rapidly restored after an oral boost and additional feeding of transgenic potato in an interesting variation of the strategy outlined above. The cholera toxin, subunit B, and A2 genes were each fused to different antigenic genes and then used to generate transgenic potato plants. To create these two fusion proteins, a 22 amino acid epitope from murine, mauve, rotavirus, introtoxin, NSP4, which fused to the C-terminal end of the cholera toxin, subunit B protein, and the intratoxigenic E. coli fimbrial colonization factor CFA by I was fused to the N-terminal end of the cholera toxin subunit A2 protein. Normally, the A2 peptide links the A1 peptide, which has the toxicity, which the subunit B peptide, which has the binding activity. Transgenic potatoes that express the two fusion proteins were fed to mice, which generated antibodies against the cholera toxin subunit B protein. Murine rotavirus, introtoxin, NSP4, and E. coli fimbrial colonization factor CFA by I and were protected against rotavirus-caused diarrhea. This approach holds great promise for the development of inexpensive and readily available vaccines for a wide range of diseases. It's been estimated that sugar toxin-producing strains of E. coli cause approximately 100,000 cases of hemorrhagic colitis a year. About 6% of those infections produce severe complications, including kidney failure. Similar to cholera toxin, the sugar toxin contains one A subunit, which encodes the catalytic toxin activity per 5B subunits, which act together to bind to cellular surface receptors. To develop an oral vaccine against type 2 sugar toxin, type 2 is responsible for the most severe disease in humans. The genes for a genetically inactivated version of the sugar toxin A and B peptides were both cloned and expressed in tobacco cells to test the ability to transform tobacco plants that synthesize the modified sugar toxin to produce mice against the toxin. Scientists infected mice with sugar toxin producing strains of E. coli. Before the introduction of the sugar toxin producing bacteria, some of the mice were fed leaves from transgenic tobacco plants, expressing the inactivated sugar toxin once a week for 4 weeks, while other mice were untreated. 1 week after the introduction of the toxic E. coli strain, by contrast, all the mice that were not fed the antigen-producing tobacco had died. By contrast, 2 weeks after treatment, the toxic E. coli strain, all of the early vaccinated mice were still alive. This experiment serves as a proof of the concept that oral administration of the inactivated sugar toxin is highly effective means of protecting animals against sugar toxin producing E. coli. Of course, for a human oral vaccine, a more suitable host plant such as tomato or banana will be desirable. Plant yield. In nearly every instance where plants are utilized, it is desirable to optimize the yield of the plant or its components. Theoretically, increases in plant yield may be achieved in a number of ways. For example, preliminary studies are directed towards introducing the C4 pathway of photosynthesis in plants that are normally used. The C3 pathway have been imported. Plants have been engineered to take up iron from the soil more efficiently. The supply of oxygen to plant cells has been enhanced, thereby increasing the rate of plant growth, and the lignin content of some trees have been decreased, making it easier to isolate the cellulose. The work of increasing plant yields is still in its infancy. Nevertheless, based on what has been achieved to date, the various strategies are likely to be fruitful. Increasing iron content. Plant growth is often limited by the availability of iron. Despite the abundance of this element on the earth's surface, plants have difficulty obtaining enough iron to support their growth because the iron in soil, especially in alkaline soil, is largely present as insoluble ferric hydroxides, which cannot be readily transported into cells. To solve this problem, fungi, bacteria, and plants secrete small specialized iron-binding molecules called silrophores that scavenge iron Once bound, the insoluble iron citriphil complex is taken up by specific receptors on the surface of these organisms. After reduction to the ferrous state, the iron is released from the citriphore. Citriphores are small molecular weight molecules, usually less than 1 kilodalton. With 3 functional or iron-binding groups connected by a flexible backbone. Each functional group presents 2 atoms of either oxygen or less commonly nitrogen that binds to iron. In chemical terms, the functional groups of bidentate and trivalent ferric iron can accommodate 3 of these groups to form a single coordinate complex. The functional groups of microbial citriphores are usually either hydroxymates or catecholates. Other functional groups include carboxylate moieties such as citrate and ethylene diamine. Plant citriphores, on the other hand, are linear hydroxy and amino substituted amino carboxylic acids such as mugenic acid and avonic acid. Other functional groups include carboxylate moieties such as citrate and ethylene adipine. Plant citriphores, on the other hand, are linear hydroxy amino substituted amino carboxylic acids such as mugenic and avonic acid. Since rice plants secrete only very small amounts of mugenic acid and are highly susceptible to growth inhibitions from iron deficiency, it was reasoned that increasing the amount of mugenic acid should enable the plant to take up more iron and hence increase the yield of the plants. Mugenic acid is synthesized in several steps. For the amino acid L-methionine, to increase the amount of mugenic acid, rice plants were transformed using agrobacterium with an 11 kb fragment of polygenomic DNA containing 2 NAT genes, NAT-A and NAT-B, encoding the subunit of the enzyme nicotinamide aminotransferase. Both these genes were under the transcriptional control of the native promoters, and the resulting transgenic rice plants, the pattern of expression of the 2 proteins was the same as in barley expression. Roots was very low in the presence of high levels of iron. It was high in the presence of low levels of iron. After 16 weeks of growth in an alkaline soil, this showed dry weight and grain yield of the transgenic plants were more than 4 times in those of the transgenic plants, and enormous differences in yield. Hopefully this spectacular gain will be realized in the field as well as under controlled laboratory conditions. Altering lignin content after cellulose. Lignin is the second most abundant organic compound on Earth. Depending on the species of tree, it accounts for approximately 15 to 35% of the dry weight of wood. Lignin is important in the mechanical support of trees and in the defense against pathogens. It is a major impediment in obtaining the cellulose, that is to say, needed by the pulp and paper industry. Releasing cellulose requires harsh physical and chemical treatment. In the United States, over 80 million tons of wood pulp is produced annually. This process, which removes nearly 30 million tons of lignin from wood, consumes enormous amounts of energy and chemicals. In addition, high levels of lignin decrease the nutritional value of forage groups that are used for cattle feed. Lignin is synthesized by the oxidative polymerization of one of 3 hydroxysennyl alcohols, be coumaryl, conifero, or silapo alcohol. One approach to reduce the level of lignin produced by quake and aspen trees, Populus triminoides, and to make it easier to harvest the cellulose, one of the steps in the biosynthesis of lignin was altered. The enzyme for fucomerate coenzyme A ligase catalyzes the conversion of fucomerate to fucomerate coenzyme A. The latter compound is a precursor of both flavonoids and lignin. Moreover, there are two isoforms of enzyme for coumarate. Coenzyme A ligase. One isoform is expressed in the epidermis of stems and leaves and is presently and probably involved in flavonoid biosynthesis. The other isoform is expressed in differentiating xylem and presumably functions mainly in lignin biosynthesis. The gene for the isoform expressed in xylem was isolated, and an antisense version of the gene was placed under the transcription control of the 25S promoter and introduced into aspirin by atomophages mediated transformation. Transformation aspirin of trees exhibit up to 45% decrease in lignin content, and at the same time as much as a 15% increase in cellulose. In addition, the transgenic trees were about 25% larger and had thicker stems and larger leaves than the non-transgenic trees. The altered lignin and cellulose content of the transgenic aspirin should make it easier to extract cellulose during pulp and paper manufacture. Recently, a considerable amount of effort has been directed to finding ways to efficiently utilize plants to produce renewable biofuels. Some of this research is focused on converting materials such as corn stover, wheat straw, grasses, and wood byproducts to glucose and then to ethanol. However, with all of these compounds, it is necessary to pre-treat the substrate under harsh conditions to remove the lignin and make the cellulose and hemicellulose more accessible to hydrolytic enzymes. These pre-treatment steps are generally costly in terms of materials and energy, and pre-treatment steps are generally costly in terms of materials and energy, and often the harsh conditions employed make it much more difficult to enzymatically digest the cellulose. However, by reducing the lignin content of plant tissues, it may be possible to employ mild pre-treatment or even no pre-treatment prior to enzymatic digestion. In fact, by constructing transgenic alfalfa plants that contain an antisense version of the gene for the enzymatic schimiate hydroxycinnamol transferase, which catalyzes one of the steps in the biosynthesis of lignin, it was possible to reduce the lignin content of the plant by about 50 to 70%. Interestingly, the transgenic plants that had the lowest lignin content also had the highest carbohydrate levels, suggesting that the plant had somehow compensated for the reduction in lignin. Moreover, when the lignin levels are low, the cell wall carbohydrates are more accessible to cellulose and hemicellulose degrading. Enzymes, importantly, since the biochemical steps that lead to the synthesis of lignin are highly conserved across the plant kingdom, it may be possible to apply this approach to a variety of plants so that, for example, corn may be engineered to continue to be a food source for other animals and humans, while the parts of the corn plant that are not consumed may be converted into biofuels. Erect leaves. Conventional varieties of wheat and rice allocate a considerable amount of their resources to the growth of vegetative tissues. On the other hand, semi-dwarf varieties developed by the conventional breeding strategies that were part of the Green Revolution allocate more of their resources to grain. In addition, semi-dwarf varieties are more resistant to damage from wind and rain and produce much less residual waste biomass. However, to feed an ever-increasing world population, it is necessary to engineer plants to produce even higher grain yields. One way to do this is to develop dwarf strains of rice and wheat that accumulate even less vegetative material and put on more resources into increasing the grain yield. Scientists have approached this approach by using genetic engineering to manipulate the levels of some plant hormones, most notably gibberellins and brasinosteroids. Brasinosteroids include more than 40 different plant polyhydroxylated steroid derivatives. The effective variety of plant processes typically at low concentrations, including cell elongation, vascular development, and stress tolerance. By genetically engineering rice plants to have a decreased level of brasinosteroids, scientists were able to increase the grain yield The crop was planted at a high density. The rate-limiting step in the synthesis of brassinosteroids is the hydroxylation of carbon-22. In rice, the hydroxylation of C22 is controlled by two differently regulated genes. One gene called the OSTWOF1 contributes primarily to brassinosteroid synthesis in shoots. The other gene primarily controls brassinosteroid synthesis for leaf inclination. And initially, a collection of transposon mutants of rice was screened for an erect leaf phenotype. That is, plants on which the leaves did not lean over but stood more or less straight up. One of the mutants that was selected was characterized in detail and found to have a mutation in the OSTWOF4 gene, mutation of the other C22 hydroxylation gene, or mutation in both genes yielded small plants that did not exhibit the erect leaf phenotype. Since the mutation that conferred the erect leaf phenotype on plants was created by transposon mutagenesis, it was relatively easy to isolate and subsequently characterize the transposon labeled gene. Importantly, when plants that showed the erect leaf phenotype were planted at a high density, they produced about 20% more rice than wild-type plants planted at the same density. This increase was attributed to the reduced shading and hence the greater amount of photosynthesis of the lower leaves of the mutant erect leaf plants. Having identified the gene whose activity is critical to increasing rice yield, it should now be possible to attenuate the activity of the gene using either antisense constructs or RNA interference. Moreover, it is expected that any approach that works in rice should be equally effective in other plants. Increasing oxygen content. Oxygen is an essential substrate for plant respiratory metabolism, and in fact, the amount of available oxygen may limit the number of plant biochemical reactions, since it is impractical to increase the external oxygen supply. To the plant. One way to increase the oxygen concentration inside a plant is to provide the plant with a protein that can sequester oxygen, and provide it where it is needed. The gram-negative bacterium Bacillus produces a dimeric hemoglobin that binds oxygen tightly and allows the bacterium to proliferate under oxygen-limited conditions. The oxygen encoding this hemoglobin has been isolated and expressed in a variety of bacteria, increasing the growth rate, the final cell density, and the yield of cloned foreign genes. When the Bacillus hemoglobin under the transcription control 35S promoter was introduced into tobacco by entomophages-mediated gene transfer, transgenic plants produce 100% more dry weight than transgenic plants. Seed germination time was reduced from 6 to 8 days to 3 to 4 days in transgenic plants, contained approximately 35% more chlorophyll and 34% more nicotine than transgenic plants. It is thought that the Bacillus hemoglobin increases the availability of oxygen and or energy in the cell. For example, synthesis of one more molecule of chlorophyll from glutamate 4 requires 4 moles of oxygen and 2 molecules of ATP. Similarly, the last step in the synthesis of nicotine, the conversion of nicotinic acid and methyl delta prime pyrrolinium salt to nicotine, is catalyzed with oxygen-dependent enzyme nicotine synthase. While this sort of genetic manipulation raises more questions than it answers, the possibility of increasing plant yield by manipulating the oxygen concentration is quite exciting. You're into the topic of phytoremediation. Phytoremediation is basically defined as the use of plants to remove, destroy, or sequester other substances from the environment. So phytoremediation of metals and other inorganic compounds may take one of several forms. The last step in the biosynthesis of nicotine. take a screenshot of this is pretty important. Phytoextraction, the absorption and concentration of metals from the soil into the plant, the rise of filtration, the use of plant roots to remove metals from effluents, phytostabilization, use of plants to reduce the spread of metals in the environment, phytovitalization. So we know the three terms. We have phytoextraction, rise of filtration, phytostabilization, and phytovolatilization. The uptake and release into the atmosphere of volatile materials such as mercury, arsenic containing compounds. A number of plants that can naturally accumulate large amounts of metal have been identified. These plants are called hypermetals. Even hyperaccumulating plants attain only a small size, that is, high concentrations of metals inhibitory to grow the plants, even those plants that are capable of hyperaccumulating metals. Depending upon the amount of metal at a particular site and the type of soil at that site could take many years to completely remove the metal from the soil and remediate the site, even with hyperaccumulating plants. Since this is too slow for the practical application, scientists have undertaken to engineer plants for more efficient metal phytoremediation. In general, plants sequester toxic pollutants in places where the toxins can do the least harm to plant cellular processes. Those pollutants are typically accumulated in vacuoles or cell walls. The uptake and accumulation leaves of inorganic contaminants with toxic effects are desirable properties for phytoextraction. To this end, plants can be engineered to have high levels of transporters involved in the uptake of inorganic pollutants from the xylem into the leaf symplast and from the cytosol into vacuoles. To engineer plants that can accumulate great amounts of lead and cadmium toxic metals that contain a wide variety of environments, the yeast gene YCF1 was used to transform Aethaliana plants. The yeast YCF protein is a member of the ABC transporter family of proteins, and when it is expressed in Aethaliana detoxifies metals, which are taken up by the plant by transporting them to plant cell vacuoles. Despite the fact that the YCF protein is expressed at a relatively low level in transgenic plants, the protein cannot be detected by immunological assays of plant cellular extracts. It can nevertheless effectively sequester metals into the vacuoles. Transgenic plants may, in the presence of high levels of either lead or cadmium, have nearly 4 times as much metals in their vacuoles and about 2 or twice as much metal overall throughout the plant as non-transformed plants. The model system is an important first step in developing transgenic plants such as poplar trees that can remove metals from contaminated soils. Organic forms of mercury, especially methyl mercury, are holotoxic to plants and animals. In plants, these compounds inhibit electron transport and the photosynthesis, both the chloroplast functions. At the present time, there are no simple and inexpensive procedures for removing mercury from the environment. However, it is possible to engineer plants with bacterial genes that encode enzymes that can detoxify organic forms of mercury. Mercury-resistant bacteria detoxify organomercurials by producing 2 enzymes, organomercurial lyase, which catalyzes the conversion of the organomercurial to the less toxic inorganic species, Hg2, and mercuric ion reductase, which catalyzes the reduction of Hg2 to the volatile and less reactive elemental form. HGO, figure 20.3, enzymes are encoded by the mauve and mauve genes, respectively, the plasmid-bound mercury resistance operon, expressing the mauve genes in plants by transformation of the nuclear genome provides some protection against the toxic effects of mercury. However, the operon containing both genes may be expressed in tobacco, chloroplasts. In a single transformation event, obviating concerns about possible positioning effects, codon usage, or transmission of the foreign genes to other plants via pollen. Moreover, chloroplast expression of the mer genes leads to a higher level of protein expression and partially protects the plant from inhibition of electron transport and photosynthesis. Having demonstrated the efficacy of this approach with tobacco, it may be useful to fine-tune the system to increase the ability of plants to detox from mercury. Also, it may be advantageous to introduce the mer operon into other plants and more extensive root systems that are better suited to take a mercury from the soil. Phytoremediation of organic compounds may occur by phytostabilization, by phytostimulation, the stimulation of microbial biodegradation around the roots of plants, or by phytotransformation, the absorption and degradation of organic contaminants by the plant. A number of different types of plants are effective at stimulating the degradation of organic molecules in the rhizosphere that is around the plant roots. I think we're talking about rhizofiltration. Typically, these plants include many common grasses, as well as crop species, have extensive and fibrous roots which form an extended rhizosphere. In addition, several varieties of trees can take up and degrade some organic contaminants. Plants with the phytotransformation activity may contain natural reductases which are used for degrading TNT, trinitrotoluene, and other natural aromatics, dehalogenases for the degradation of chlorinated solvents, and pesticides and lactases that can degrade anilines such as trans-amylotoluene. However, there is less incentive to engineer plants to be more efficient degraders of organic compounds, since many plants can already do this effectively, and two, selecting and engineering soil microbes that live in the vicinity of plant roots that provide a simpler means to the same end. And now for the summary. The expression of foreign genes in plants makes it possible to produce a wide range of new varieties. Plants with new colors have been developed, nutrition contents of crops have been enhanced, discoveries from potatoes have been prevented by genetic manipulation, sweetness of some plants have been augmented. Plants have been developed to act as factories or large-scale production of important foreign proteins such as antibodies and therapeutics, and plant yield has been increased by increasing the iron and oxygen content, by modulating the lignin content, and by modifying the response of the plant to present steroids. Also, plants are being utilized as components of phytoremediation protocols, designing to remove contaminating metals and organic compounds from the environment. Plant nutrition content may be improved in a variety of ways. The amino acid content, specifically methionine and lysine, can be increased. The lipid composition can be modified to suit the intended use of the oil. Pathways for the synthesis of vitamin E and precursor vitamin A have been engineered, and plants with increased levels of available iron have been created. As bioreactors, plants can produce on laboratory scale monoclonal antibodies and antibody fragments, the polymer polyhydroxybutyrate, which can be used to make a biodegradable plastic-like material, and a number of potential therapeutic agents found in transgenic plants have also been used as edible vaccines, an approach that could result in a wide range of new and inexpensive vaccines. We just have a few review questions. How can plants be genetically manipulated to produce flowers with unusual colors? How can soybeans be genetically manipulated to increase lysine content? How can vitamins be overproduced in rice plants? How would you engineer soybean plants to overproduce long-chain omega-3 and omega-6 fatty acids? What is the effect of increasing the level of oxidized glutathione within a plant? How would you genetically manipulate a plant to do this? How can the level of bioavailable iron in plants be increased? How would you increase the amount of amylose compared to maltopectin in potato starch? Would you increase the amount of maltopectin compared to amylose? What is the advantage of performing these manipulations? How does antigen expressed in transgenic plants act as edible vaccine? Would you engineer an edible vaccine directed against E. cholerae? How can a plant's oxygen levels be manipulated? How does manipulation of plant's oxygen levels affect yield of that plant? How would you engineer rice plants to overproduce tetrahydrofluoride? Why would you do this? Why is it necessary to genetically engineer soybean plants to have a small amount of phytate in their seeds when low pattern mutants may be selected following conventional mutagenesis? Briefly describe a vector system. Why are plants an attractive host system? Why is phytate? Describe a strategy for developing plant vaccine. How can the lignin content in trees be decreased? How can manipulation of levodopa and steroids be used to increase plant yield? And how can plants be genetically modified to increase the effectiveness of phytoremediation? That ends the lecture here. I'll meet you in the next lecture soon. Thank you. Best wishes. Bye.